culture, how you grow ants in the colony and from a single queen. So today's presentation is coming from what I have tried and found some error and tried to fix it over the 2 years. I have read some literature about it. I have found some videos from the ant hobbyist. I've tried different things, and now we kind of have a proper systematic way to culture ants. So I'm going to talk about that. So first of all, why should we culture ants? Because ants are those kind of a animal system which has potential for different kinds of experiment, different kinds of research, and it's not that difficult to get a setup in the lab. So it's very good system, control system where you can do like incredible experiments on them. So you can do some of the examples are like you can do developmental studies, like you just observe ant colonies over the time with a particular constant environment, and you see how they develop over time. For different species, you can compare different species developmental rate, what kind of food is affecting them, helping them to grow. You can also do some manipulative studies where you change different factors that can affect their growth. For example, you can change temperature, you can change the lighting conditions, you can change the food condition, and you can see how that affects the development and growth of the colony. And you can also see behavioral you can also do behavioral studies, so you can see individual roles inside the colony and outside the colony. So most of the time the foragers who are usually go outside the colony, so you can see what kind of roles they play inside the colony. Even there are studies where people have tried to understand trophallaxis, which like ant to ant feeding, because foragers bring food to the colony and they feed other workers as well as brood like larvae and the queen. So you can see what kind of networks are there between who feeds whom, and then how does foragers can be helpful in the whole overall growth of the colony. So these kind of behavioral studies and network studies also has been done with the lab colonies. So yes, it has extremely good potential for studying different aspects of the science. Now I'll just explain what kind of basic ant colony structures are there. Like what is the structure of the colony? So colony has properly reproductive division of labor, where only certain individuals perform reproduction and other individuals do not take part in reproduction. So whoever takes part in reproduction are generally queens and males, and the workers are generally non-reproductive. So the sole task for queens and male is to reproduce. So males die after mating with the virgin queens, and queens who can lay eggs and then can be the like founding individual of the whole colony, and it can live up to years and years, like decade or more. So a single queen has a potential to like give millions of eggs over the years. And workers are the ones who perform all the non-reproductive tasks in the colony, so finding food for the colony, taking care of the brood, uh, um, then cleaning the nest and protecting the nest from the enemies. So these all tasks are performed by workers. Now yeah, I just want to tell that you guys can stop me anytime, any point, if you have any question or some confusion, so that's properly fine. So, what is the life cycle of the ant, or like what are the brood stages are there? So, when we have a single queen in the colony, queens generally mate with the male, and after mating, it can store the sperm in a organ called spermatotheca, and then it can store that sperm, and it can use the sperm to fertilize its egg, and then it can lay the eggs, which will develop into larvae. So larvae stage is the most crucial stage in the ant's life, because it is the phase where they'll get all the nutrition from the mother or their sibling, like sisters. And what kind of nutrition you get during the larval phase decide what kind of work you'll be doing, what kind of task you'll be doing, or what kind of caste you'll develop into. So there are different castes in the ant colony. Some are workers, some are soldiers, major soldiers, minor soldiers, depending on what kind of species is that. and what kind of nutrition you are getting during the larval phase will decide what kind of morphological structure or caste you'll become. So that's why it is very important and crucial phase in the ant's life. Now, larvae will develop into pupae, and then pupae will eclose and become worker. So most of the workers will become most of the pupae will develop into worker. But yes, there will be some pool of the eggs, larvae, and pupae that ultimately will become males, and some, like very few, which are called royal eggs, and which is generally larger in size and get properly like very different nutrition from the other larvae, and that will eventually become the future queen. So this is the life cycle which is common in all of the species. Now, so this actually tells us that if we have a single queen, we can develop the whole colony from a single queen. We don't need separately the brood or separately workers to build a colony in the lab. So that way, it is very efficient to find the queen. So only question or only issue is finding a queen. So how to find a queen? So first of all, we need to understand what is the where when is the mating season and where we can find them, and what time is important to find them. What time we can actually find them. So season is generally pre-monsoon showers. So when monsoon starts, before that we see that there will be one or two rains that will be far apart from each other. Like it rained today and then it will rain after few days. So that initial one or two showers, you'll find high mating activity of ants. So during this time you can find queen. Now, time is generally after sunset. Why? Because once the queen is mated, it actually has very limited amount of time to dig a nest and go inside the soil. Because if it is it is delayed, it gets delayed, then there is a high chance of predation in the morning by birds. Because ants are like queen ants are very rich in nutrition, so birds can predate on them in the morning. So generally, the time is after sunset when there is a rain. And generally, I have observed that when there is a high mating activity and you find them usually after this 1 hour period window period after the mating activity. So high mating activity is when you have a pre-monsoon shower. So during the showers, you will see them near the light pole. uh, there will be so many insects flying, so there will be other insects as well as ants, like males and queens. So if you observe such place, you just have to wait half an hour to 1 hour after this mating activity subsides. You will find them on ground. So it can be on the concrete ground or loose soil near this light pole. So these are the very simple things you have to remember to find a queen. Just pre-monsoon showers, sunset, find a high mating activity, and then after that you find them on ground. Now you know all this stuff, like where to find and how to find, but how can you identify queen? So queens are generally large in size than workers. So what you usually see, the size of workers which you see roaming around anywhere, so they are generally 5 to 10 times bigger than the worker. Now this cannot be the only thing you should observe. You can rely on, because there will be different sizes of ants. So if you don't know what is the normal size of particular species workers are, it will be difficult for you to identify the queen. So what is the other aspect? So presence of wings. So in the ant colony, only males and queens have wings. Worker do not possess wing. Why? Because mating activity occurs in air. So they have to fly, and then in the flight they mate, and then they land on the ground. So only males and queens have wings. Now, if both of them have wings, how do you distinguish between males and queens? So they also have different morphology. For example, you can see here in the photo that males have very narrow and slender belly with a pointed tip of the abdomen. But queens' abdomen is generally bulkier and larger. So that is, yes, the size difference is there, but this is a particular thing where you can actually see the difference between males and females. The morphology of abdomen. This is very pointy and narrow, and queens are bulkier and larger. Now, there is one more feature which is like it is a sure thing to tell that the particular individual is queen. is the big thorax and the presence of wing scars. So if the queen is mated, it you might find them with wings or without wings. So how do you know if you find a queen without wings? It will be difficult to understand if it is a queen or soldiers, because soldiers are also larger in most of the species. So you can see, look for the scars on the thorax. So after mating, queens generally shed their wings and find the nest. So if you find a queen without wings, you can look for the scars where wings used to be present. So I'll show you one figure. So this is also a queen, but it does not have wings, it has shed. But you can see that this is a scar here where wings used to be present. So this is a particularly like a unique feature where you can 100% sure that a particular individual is queen only, not male, not worker, not soldier. So these three features are there: large size, presence of wing if unmated or recently mated. So if the queen is unmated, it will not shed its wings until it's mated. And then big thorax and presence of wing scars. Now, there are two types of queen you might find: fully claustral and semi claustral. So, fully claustral queen is the queen who were virgin and mated for the first time, and it has to find the new nest to build a colony. So, that would be the first time the queen would start its own colony. Semi claustral queens are the queens which have already established colony, but for some reason it has separated from the actual colony, or its colony has died and it has to find a new place to uh, make a nest. So these are semi claustral queens, which is definitely will be without wings. But fully claustral, you may find them with wings or without wings. So this is the difference between fully claustral and semi claustral. Fully is the first time nest maker, and semi claustral is they have already nest but now has to make a new nest, or somehow they are out of the nest. Now, once you know that you have found the queen, you have identified the queen, what should you do if you come across the queen? So first thing is, a basic setup is test tube setup. So during our first time, we tried many different things to make a nest, but then ultimately we have found that nest setup is somewhat standardized and it is working for most of the species. So in the test tube, simple normal test tube with a water, you just have to plug the cotton in the water in a way that it should not leak outside this part. The cotton should be wet. You should fill the water up to like 50 to 60%, and there will be little space for the queen and upcoming workers to stay there. And you plug the cotton to make them stay inside. You can directly capture the queen into the test tube. So most of the time, it's better if you just don't pick the queen with the fingers or forceps or anything. It would be better if you just capture them inside the test tube directly. So you can just put the test tube on the top of the queen. It will try to find the exit. If it is not finding the exit, it will try to crawl up the test tube, and you just plug the cotton. That is the easiest way, like very less stressful way to capture the queen. If it is difficult, you can nudge it with the cotton and then plug the cotton to trap it inside. This cotton should not be very tight. That won't allow the air to like travel in and outside. So it should be. It's very simple setup. Then you can write all the different things of capture, where you have captured, on which date you have captured. All the things you can put on the test tube, and that's very easy to capture the queen. Now, these are some of the queens we have captured this year. Uh, these all species are Camponotus, and this the smallest one is the Phaidol sulcaticeps. So you can see the size difference, right? So I was telling you about the queens are larger. So this is also a queen. This is also a queen, but the size difference is too much. So it's not like this is a worker, but this is a different species which is smaller. So obviously, its queen is also smaller. So you cannot just rely on the size of the queen. You have to check for the other features. Now, I don't have any particular zoomed in photo for a particular queen. Otherwise, I would have shown you the scar on the thorax. But you can see the thorax is larger compared to the workers. So, and there you can find the scar. Now, what is the initial requirement? So you have captured the queen. Now you have it in the test tube. What should you do and where you should keep it? So first thing is, because they nest under the ground. Under the ground there is no light. So first thing is they require dark place. So you just keep the test tube in the dark place with no disturbance. So there should be like no light fluctuation. There should be no noise in that environment. So that would all cause some stress in the queen, and it might not lay eggs. So you should find a dark place with no disturbance. Now, what should you feed in the initial phase? Now, founding queens are the claustral queens, which is first time has mated and will start the colony. They do not require food. So in initial phases, it will just use, because it has already shed wing muscles. So now the wing muscles, it will digest inside, and it will produce a food, and it will feed it to the initial workers, initial larvae. So you do not have to feed the founding queens in the initial phase, like when until the first workers arrive. So when you have like 3 4 workers, then you start giving them food. Otherwise you don't need to give them food. Now you should not check it like very frequently. That would also cause them some stress. So you should keep it undisturbed for at least a week or 10 days. You If you have to check for the condition of queen, the setup and everything, you can check once in a week or 10 days. And then you can provide them food when the workers arrive. And specifically two main nutrition they require is carbohydrate and protein. So basically, for carbohydrate, if you can give them honey solution, you can give them sugar solution. And for protein, you can give any crushed insects or some artificial food like cat food and all. But it's better to give them something that they can find it in natural environment. So basically, some insect larvae or some crushed insects. Most of the time, people feed them with crickets. We feed them in with moth, the small moth. And the temperature requirement is 26 to 29 degree. That is also depending upon species. So all the tropical species is thrives better up to like 28 to 29 degree, but some temperate species also go 25 to 26 degree centigrade. So this should be initial requirement. And now what should you record when you have a colony? So these are only basic stuff. So you should fix a day and time where you should check what kind of colonies developmental stages. You should do it over the longer period of time, because ants have some of the ants have very slow development rate, some are very fast. So you should count how many numbers of eggs, larvae, pupa and worker in the each observation. Now counting eggs is very difficult because they are very small, and they are generally clubbed together. So eggs are not scattered. Even in your setup, if you found the eggs are scattered, that means that egg will not hatch, and for some reason queens have discarded those eggs. So counting eggs is difficult. If you can count it if you have, but then you have to remove the eggs, and you have to keep it separate in the separate setup or under the microscope. Now there will be some phases where you find that queens have laid eggs, and when the next time you check, you'll find them there are no eggs. So queens also feed on their eggs. So queens can decide which egg they want to fertilize by sperm, and uh, they can lay different kinds of eggs. So they can fertilize the eggs, and they can lay the eggs that eventually become workers that are females. If the queen has laid non-fertilized egg, that will eventually become males. But queens can also lay trophic eggs. So trophic eggs is she'll lay eggs and she will eat it. So it's for a own nutrition. So if you find any such incidents where the eggs were present one time and the next time you don't find eggs, that means they were trophic eggs and queen might have eaten them. And then next thing is you provide food, and you should check, like you should note what kind of food you have provided and which date and all. So that will help you understand what food is working for the particular species you have, and what food is not working, or their preference for the food you can see. So these are the observation you can record. If you do not have particular particular goal for the colony. Now, yes, also you can write about condition of the setup, because you'll find them, there'll be sometimes leakage of the water outside the plugged cotton, or you'll find some discoloration on the cotton, you'll find some fungal infection or something. So you should observe that condition of the setup, is if it is good or not. Now, what can be the common problems you can come across while this ant rearing? So first is desiccation. So when I explained the test tube setup, I told that cotton should be wet where you plug it with the water. So if it is dried up, then ant would not get water. So water is important for the ants to drink from, and also as a moisture, because the brood requires moisture. If the environment is too dry, the eggs and larvae won't develop and it would dry up. So you should see that the cotton should be wet all the time. If it is dried up, you should shift the colony into some other setup, or you should make it wet, like inject some water into the cotton to avoid the desiccation. If it is too high, if it is high disturbance, so if you keep on checking on them, and if the disturbance is too high, so that can make the queen stressful, and then it won't lay eggs. So in most of like in our setup, I used to check them on once in a week only, and I just try to take the tube out and in my hand only when the workers are there. I used to just count the eggs and larvae while keeping them inside only, because when you keep it out, your hand will shake, and that would also cause the queen in stress. So if the disturbance is too high, queen will not lay eggs. uh, Wrong identification of the queen type. So if you don't know if the queen is mated or unmated, you might have captured mated queen with wings, sorry, unmated queen with wings, it will not lay eggs. But I have seen unmated queens also laying eggs, but those all eggs were uh, definitely virgin, because she doesn't have sperm in her spermatotheca. So all the queens were virgin, and they have developed into males. So when you see the queen is winged, and the eggs and larvae ultimately developed into males, the queen is definitely virgin. So in such cases, you can release the queen, or you can just keep on like giving food, and you can just observe what happens. Yes, unmated queen, as I mentioned. And if you don't know which species the particular queen is, that might also lead to some problems in their development, because you don't know the basic needs for a particular species, the what kind of space they require, what temperature they thrive in, and what kind of food they rely on. So that can also cause problem into their development. And this is the biggest issue even I am facing nowadays also, is the fungal growth or mold growth inside the colony. But that would not happen in the initial phase, but it can happen at any stage in the colony development. So, and I have not found yet the way to stopping the fungal growth. Most of the time I see if the fungal is growing too much, and the numbers, like workers number, is going down, like mortality is high, then I shift them to a separate fresh setup. Because we pick up the queen from the ground and all, so queen might also have some kind of spores of the fungus on their body. So I also find this problem in my colony, like in our lab also. So these are some common problems, but yes, it does not mean that you cannot do it. I'll share you my success story. Our lab has like very good, like I don't know if it is very good, but we are very happy with whatever we have achieved so far. So we collected 22 queens in 2024 during pre-monsoon showers of three species: Meranoplus bicolor, Solenopsis invicta, that is fire ants, and Paratrechina longicornis, which is very common black garden ant, you can see, and Meranoplus bicolor is furry ants. So out of all these species, there were like 5 to 6 virgin queens, and so far the other species like Solenopsis and Paratrechina didn't survive, But out of this 22, we have now 4 colonies of Meranopus bicolor. This furry end, you can see the picture behind. We have 4 colonies with a number of workers 35, 42, 76, and 103. So these 2 colonies are doing good. All these 4 colonies are now 2 years old, but for some reason, these 2 colonies do not have good development, good growth in the colony. But even though I've given them similar amount of food, kept them in similar amount, similar setup, similar temperature, and all, the worker number still differs. Uh, yes, so this was in 2024. The picture I showed you earlier for Camponotus, those queens we collected this year. So we collected 48 queens this year. It belongs to 4 different species: Camponotus compressus, which is the picture you can see behind; Phaidol sulcaticebs, that is big-headed ants; Camponotus rufoglaucus, that is velvet sugar ant; and Solenopsis geminata. Solenopsis geminata is also a species of fire ant. So these 3 species we collected from Gujarat, and this one is from Kerala. uh, we have like maximum right now, all out of 48 we have 46 queens. 2 queens have died, and out of 46, 2 queens have not laid eggs. All the other queens have laid eggs, developed into larvae, pupae, and we have workers for all of the species now. So yes, we have like around 50 colonies in our lab right now, and we are very happy with it. um, so I will just show you some of the pictures. So this is Meranopus bicolor. So I've kept them in this test tube setup. So initially I've kept them in smaller test tube, and when the number increased, I have given them bigger test tube. And I have connected. So initially I used to give food in the test tube itself, but now I've connected them with some external area where they can come out and go inside through this small tube. They can come out, and I just put food inside this box, inside this container, and then they can take the food inside. They can feed on and do this stuff. So in this condition, I can clearly see that number of foragers which comes out and find food is somewhat proportional. So you can always see that not all of the individuals come out. Only some part of individuals, like out 30% of individuals of the total number, would just come out and grab the food and go inside. Yes, so this is the colony. I I think yes. So this is the queen, which is a little bit bigger. If you can make out the just above my blue dot, that's the queen, and the white thing you are seeing is the brood. Most of the time, you'll see larvae, and they also have tendency to group the brood into separately eggs, larvae, and pupa. They keep it separate most of the time. But yes, different species have different structure in the colony. uh, so later on, we have provided them this 3D printed uh, maze, so that this like resemble their actual nest structure inside the ground. They have different chambers where they keep them, one chamber for food, the other for the brood and stuff. So we have tried to replicate their actual nest structure uh, with some wet cotton, so they can get access to the water, and they can like place their brood if moisture is required. So ants can uh, they can sense what kind of moisture and temperature the brood requires, and that way they can shift the brood inside the nest according to the requirement. And then I can connect some external test tube, and I can give them some food. So now in the next phase, we are planning to give some bigger structure which will be connected with this nest with some tube, and we will give them some structure with some sand and some vegetation, uh, replicating the natural environment where they can come and go, and like for foraging and other activities. So that is our next phase. I hope I'll will be able to do this, and like I can share the success story with you guys very soon. Now I'll explain some of the like two of the example studies where people have done very nice work where they have reared the ants in the lab and then they performed some manipulative experiments. So this is very recent study of 2024. It is done in Leish's Niger species, and they have experimentally manipulated feeding the food types and frequency, and then observed what is the developmental rate. So their main aim is to give the supplementary feeding to the claustral colony. So as I mentioned earlier, the claustral queens does not require food in the initial phase. They just use their wing muscles to feed the initial workers. But they are just checking if we give the food to the claustral queen, does it improve the number of workers developed from the initial phase. So they have given them different types of food. They have collected some 240 queens and divided them into eight different groups. And in each group, they have given them different kinds of food and different treatments and see how it develops. And they also change the feeding frequency to like they never fed them, they once fed them, and they fed them weekly. And they also observed if you disturb them more or less, does it affect the development. So what they found is, so type of food is carbohydrate and protein, and in combination, and no food. So they found that if you feed them weekly with protein source, then it is the most effective. You'll find maximum number of pupae and worker developed out of the brood. So yes, even if the claustral queen do not require food in the initial phase, but if you feed them, that colony would develop faster. And if you disturb them, it does not significantly affect their nesting success. But this is for only Lasius niger. So queens and species have different sensitivity towards different disturbances. So that depends what kind of species you are working with. But yes, this study was very cool, so I thought I would share it with you guys. And the other study where they've change the temperature and see what is the development and survival of the particular ant species, Argentine ant. Uh, Yes, so in that they have range of temperature they have given starting from 18 to 32, and they just counted number of X larvae in pupa and their development rate. And they found that optimal temperature is 26 for this particular species, and the lowest temperature 18 and highest 32 has a developmental limit. So in this case, no eggs and larvae is developed in the high lowest and coldest and heat like most higher temperature. But there is also an interesting thing where they found that there is a state specific vulnerability. So if it is too hot or cold, it affects different stages of the ants. So if it is too hot, the eggs would not develop, but larvae and pupae would be fine. But if it's too cold, the larvae would not go into next stage. However, pupa is very resistant to the temperature. It would have similar survival rate in all different kind of temperatures. So these are just the example of study where you can manipulate different things, but there are tons of study you can do, tons of experiments you can do when you have a lab colony. You can even just the observation, because right now I am not doing any manipulative experiment with the species we have, but even the observing them and just noting down normal the number of these things, and you when you find them in different when you find them different showing them different behaviors, it's very interesting to just look at them. It's kind of a therapy. Whenever you watch ants, it's like a very cool thing to just observe them. It's very fascinating for me at least. So yes, that's it. I hope you find it very interesting, and yeah, if you have any questions, I'm happy to take it. Thank you all.